Chapter number 2. Biological classification. Since the dawn of civilization, there have been many attempts of classifying living organisms. It was done instinctively, not using criteria that were scientific but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis of classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which has red blood and those that did not. In Linnaeus' time, a two-kingdom system of classification with Plantae and Animalia kingdoms was developed that include all plant and animals respectively. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic green algae and non-photosynthetic fungi organisms. Classification of organisms into plant and animal was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of fall, mode of nutrition, habitat, method of reproduction, evolutionary relationship, etc. Classification system for the living organisms have hence undergone several changes over the time. The plant and animal kingdom have been a constant under all different systems. The understanding of what group organisms we include under these kingdoms have been changing. The number and nature of other kingdoms have also been understood differently by different scientists over the time. R.H. Whittaker, 1969, proposed a five-kingdom classification. The kingdom defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. The main criteria of classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationship. Table 2.1 give a comparative account of different characteristics of the five kingdoms. Table 2.1 Characteristics of the five kingdoms. Characters. Cell type. Cell type Monera, Protista, Fungi, Plantae, and Animalia. Cell type Monera have only prokaryotic cell type. Other have eukaryotes. Monera have cell wall, non-cellulosic, polysaccharide, and amino acid. Protista have cell wall in some. Fungi have cell wall chitin. Plantae have cellulose, zinc cell wall. Animalia have not cell wall. Monera have not nuclear membrane. Others have nuclear membrane. Monera body organization cellular. Protista body organization cellular. Fungi body organization multicellular loose tissue. Plantae body organization tissue and organ. Animalia body organization tissue, organ, and organ system. Monera mode of nutrition autotrophic, heterotrophic. Protista autotrophic, heterotrophic. Fungi heterotrophic. Plantae autotrophic. Animalia heterotrophic. The three-domain system has also been proposed that divides the kingdom Monera into two domains, leaving the remaining eukaryotic kingdom in the three domain, and thereby a six-kingdom classification. You will learn about this system in detail at higher classes. Let us look at this five-kingdom classification to understand the issues and consideration that influenced the classification system. Earlier classification system include bacteria, blue-green algae, fungi, mosses, fern, gymnosperm, and the angiosperm under plants. The character that unified this whole kingdom was that all the organisms include had a cell wall in their cells. This placed together group which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae cyanobacteria with other groups which were eukaryotic. It also grouped together the unicellular organisms and multicellular ones. Say, for example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group fungi and the autotrophic green plant, though they also shown a characteristic difference in their wall composition. The fungi has chitin in their walls, while the green plant had a cellulosic wall. When such characteristics were considered, the fungi were placed in separate kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has brought together Chlamydomonas chlorella, earlier placed in algae within plant and both having cell wall, with paramecium and amoeba which were earlier placed in animal kingdom which lack cell wall. It has put together organisms which in earlier classifications were placed in different kingdoms. This happened because the criteria of classification changed. This kind of changes will take place in future too, depending on the improvement in our understanding of characteristic and evolutionary relationship over time. An attempt has been made to evolve a classification system which reflect not only the morphological, physiological and reproductive similarities, but it also phylogenetical, that is based on evolutionary relationship. In this chapter, we will study characteristic of kingdom Monera, Protista, fungi of the Whittaker system of classification. This kingdom, Plantae and Animalia, commonly referred to as plant and animal kingdom respectively, will be dealt separating in chapter 3 and 4. 2.1 Kingdom Monera. Bacteria are the sole member of Kingdom Monera. They are the most abundant microorganisms. Bacteria occurs almost everywhere. Hundreds of bacteria are present in handful of soil. They also live in external habitats such as hot spring, deserts, snow, and deep ocean, where very few other life forms can survive. Many of them live on other organisms as parasites. Bacteria are grouped under 4 categories based on their shape: the spherical coccus, cocci, the rod-shaped bacillus, bacilli, the comma-shaped vibrium, vibrio, and the spirillum, spirilla. Figure 2.1. Bacteria of different shape: cocci, bacillus, spirilla, vibrio. Though the bacterial structure is very simple, they are very complex in behavior. Compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrate. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs, that is, they depend on other organisms or on dead organic matter for food. 2.1 Archaebacteria. These bacteria are special since they live in some of the most harsh habitats such as extreme salty area halophils, hot spring, thermoacidophils, and marshy area methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals such as cows and buffaloes, and they are responsible for the production of methane biogas from dung of these animals. 1.2 Eubacteria. These are thousands of different eubacteria or true bacteria. They are characterized by the presence of a rigid cell wall and, if motile, a flagellum. The cyanobacteria, also referred to as blue-green algae, have chlorophyll A similar to green plant and are photosynthetic autotrophs. In figure 2.2, the cyanobacteria are unicellular, colonial, filamentous, freshwater marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. They often form bloom in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in a specialized cell called heterocyst. Example, Nostoc and Anabina. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrates, nitrite, ammonia, and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume, fruits, etc. Some are pathogens causing damage to human beings, crops, farm animals, and pets. Chlorella, typhoid, tetanus, citrus canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fission. Sometimes under unfavorable conditions they produce spore. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to the other. The mycoplasma are organisms that completely lack a cell wall. They are the smallest living cells known and can survive without oxygen. Many mycoplasma are pathogenic in animal and plant. Kingdom Protista 2.2. All single-celled eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What many we photosynthetic protistants to one biologist may be a plant to another. In this book, we include chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans under Protista. Members of Protista are primarily aquatic. This kingdom forms a link with the other dealing with plant, animal, and fungi. Being eukaryotes, the protistans' cell body contains a well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia. Protists reproduce asexually and sexually by a process involving cell fusion and zygote formation. 2.2.1 Chrysophytes. This group includes diatoms and golden algae desmids. They are found in fresh water as well as in marine environments. They are microscopic and float passively in water currents, plankton. Most of them are photosynthetic. In diatoms, the cell wall forms two thin overlapping shells which fit together as in a soapbox. The walls are embedded with silica and thus the walls are indestructible. Thus, diatoms have left behind large amount of cell wall deposits in their habitat. Their accumulation over billions of years is referred to as diatomeous earth. Being treated, this soil is used in polishing, filtration of oil and syrup. Diatoms are the chief producers in the oceans. 2.2.2 Dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue, or red depending on the main pigment present in their cells. The cell wall has stiff cellulose plate on their outer surface. Most of them have two flagella. One lies longitudinally and the other transversely in a furrow between the wall plates. Very often, red dinoflagellates, example, Gonicolex, undergo such rapid multiplication that they make the sea appear red. Red tides. Toxins released by such large number may even kill other marine animals such as fishes. Euglenoid. Majority of them are freshwater organisms found in stagnant water. Instead of cell wall, they have a protein-rich layer called pellicle, which make their body flexible. They have two flagella, a short and a long one. though they are photosynthetic in the presence of sunlight. When deprived of sunlight, they have behaved like heterotrophs by predating on other smaller organisms. Interestingly, the pigment of eukaryotes are identical to those present in higher plants, example euglena. 2.4 Slime molds. Slime molds are saprophytic protists. The body moves along decaying twigs and leaves, engulfing organic material. Under suitable conditions, they form an aggregation called plasmodium, which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiates and form fruiting body, bearing spores at their tip. The spores possess true walls. They are extremely resistant and survive for many years even under adverse conditions. The spores are dispersed by air currents. 2.5 Protozoans. All protozoans are heterotrophs and live as predator or parasites. They are believed to be primitive relative of animals. They are four major group of protozoans. Number 1. Amoeboid protozoans. These organisms live in freshwater, seawater, or moist soil. They move and capture their prey by putting out pseudopodia, false feet, as in amoeba. Marine forms have silica shell on their surface. Some of them, such as ant amoeba, are parasites. Number 2. Flagellated protozoans. The members of this group are either free-living or parasitic. They have flagella. The parasitic form cause diseases such as sleeping sickness. Example, trypanosoma. Number 3. Ciliated protozoans. These are aquatic, actively moving organisms because of the presence of thousands of cilia. They have a cavity gullet that open to the outside of the cell surface. The coordinated movement of rows of cilia causes the water laden with food to be steated into the gullet. Example is paramecium. Number 4. Sporozoans. This includes diverse organisms that have an infectious spore-like stages in their life cycle. The most notorious is Plasmodium malarial parasite, which causes malaria, a disease which has a staggering effect on human population. 2.3 Kingdom fungi. The fungi constitute a unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. You must have seen fungi on a moist bread and rotten fruits. The common mushroom you eat and toadstool are also fungi. White spots seen on mustard leaves are due to a parasitic fungus. Some unicellular fungi, example yeast, are used to make beer, bread and beer. Other fungi cause diseases in plant and animal. Wheat rust causing Puccinia is an important example. Some are the source of antibiotic penicillium. Fungi are cosmopolitans and occur in air, water, soil, and on animal and plants. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in refrigerator? Yes, it is to prevent food from going bad due to bacterial or fungal infection. With the exception of yeast, which are unicellular, fungi are filamentous. These bodies consist of long slender thread-like structure called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tubes filled with multinucleated cytoplasm. These are called conocytic hyphae. Others have septa or cross wall in their hyphae. The cell wall of fungi are composed of chitin and polysaccharides. Most fungi are heterotrophic and absorb soluble organic matter from dead substrates and hence are called saprophytes. Those that depend on living plants and animals is called parasites. They can also live as symbionts in association with algae as lichen and with root of higher plants as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission, and budding. Asexual reproduction is by spore called conidia or sporangiospore or zoospores, and sexual reproduction is by oospore, ascospore, and basidiospore. The various spores are proce- produced in distinct structure is called fruiting body. The sexual cycle involves the following three steps. Number one, fusion of protoplasm between two motile or non-motile gametes is called plasmogamy. Number two, fusion of two nuclei called karyogamy. Number three, meiosis in zygote resulting in haploid spores. When a fungus reproduces sexually, two haploid hyphae or compatible mating type come together and fuse. In some fungi, the fusion of two haploid cells immediately results in diploid cells too, and however, in other fungi, Asco and Basidio, an intervening dikaryotic stage n plus n, that is 2 nuclei per cell, occurs. Such a condition is called dikaryon, and the phase is called dikaryo phase of fungus. Later, the parental nuclei fuse and cell become diploid. The fungi from the fungi form fruiting bodies in which reduction division occurs, leading to formation of haploid spores. The morphology of mycelium, mode of spores formation, and fruiting body form the basis for the division of kingdom into various classes. 2.3.1 Phycomycetes. Members of Phycomycetes are found in aquatic habitat and on decaying wood, in moist and damp places, or as obligate parasites on plant. The mycelium is aseptate and coinocytic. Asexual reproduction takes place by zoospore motile or by aplanospore non-motile. These spores are endogenously produced in sporangium. A zygospore is formed by fusion of two gametes. These gametes are similar in morphology, isogamous, or dissimilar, anisogamous or oogamous. Some common examples are Mucor, Rhizopus, the bread mold mentioned earlier, and albigo, parasitic fungus on mustard. 3.2 Ascomycetes, commonly known as sac fungi. The Ascomycetes are mostly multicellular, example penicillium, or rarely unicellular, example yeast Saccharomyces. They are saprophytic, decomposer, parasitic, or coprophilous, growing on dung. Mycelium is branched and septate. The asexual spore are conidia, produced exogenously on special mycelium called conidiophores. Conidia on germination produce mycelium. Sexual spores are called ascospores, which are produced endogenously in in sac like asci, singular ascus. These asci are arranged in different type of fruiting body called ascocarps. Some examples are Aspergillus, Claviceps, Neurospora. Neurospora is used extensively in biochemical and genetic work, and many members like morels and truffles are edible and are considered delicious. 3.3 Basidiomycetes. Commonly known form of Basidiomycetes are mushroom, bracket fungi, or puffballs. They grow in soil, on logs and trees stumps, and in living plant bodies are parasites, that is rust and smuts. The mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organ are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strain on genotypes. The resultant structure is dikaryotic which ultimately gives rise to basidium. Karyogamy and meiosis takes place in the basidium, producing four basidiospores. The basidiospores are exogenously produced on the basidium. Plural is basidia. The basidia are arranged in fruiting bodies called basidiocarps. Some common members are agaricus, mushroom, Yastilago, ismut, Puccinia, rust fungus. 3.4 Deuteromycetes, commonly known as imperfect fungi, because only the asexual or vegetative phases of these fungi are known. When the sexual forms of these fungi were discovered, they were moved into class they rightly belong to. It is also possible that the asexual and vegetative stage have been given one name and placed under Deuteromycetes, and the sexual stages another and placed under another class. Later, when the linkages were stabilized, the fungi were correctly identified and moved out of Deuteromycetes. Once perfect sexual stages of members of Deuteromycetes were discovered, they were often moved to Ascomycetes and Basidiomycetes. The Deuteromycetes reproduce only by asexual spore known as conidia. The mycelium is septate and branched. Some members are saprophyte or parasites, while a large number of them are decomposers of litter and help in mineral cycling. Some examples are Alternaria, Coletrotychum, and Trichoderma. 2.4 Kingdom Plantae. Kingdom Plantae includes all eukaryotes chlorophyll containing organisms commonly called plant. I think we should discuss Plantae and Animalia in next another chapters. So skip it. 2.6 Virus, viroids, prions, and lichens. In the five kingdom classification of Whitaker, there is no mention of lichen and some acellular organisms like virus, viroid, prions. These are briefly introduced here. All of us who have suffered the ill effect of common cold or flu known what effects virus can have on us. Even if we do not associate it with our condition, virus did not find a place in classification since they are not considered truly living. If we understand living as those organisms that have a cell structures, the viruses are non-cellular organisms that are characterized by having an inert crystalline structure outside the living cell. Once they infect a cell, they take over the machinery of the host cell to replicate themselves, killing the host. Would you call virus living or non-living? Virus mean venom or poisonous fruit. Dmitry Ivanovsky, 1892, recognized certain microbes as casual organisms of the mosaic disease of tobacco. These were found to be smaller than bacteria because they passed through bacterial-proof filters. M.W. Wizarnik, 1898, demonstrated that the extract of the infected plant of tobacco could cause infection in healthy plant and named the new pathogen virus and called the fluid as Contagium vivum fluidium, infectious living fluid. W.M. Stanley, 1935, showed that the virus could be crystallized and crystals consist largely of proteins. They are inert outside their specific host cells. Viruses are obligate parasites. In addition of protein, virus also contain genetic material that could be either RNA or DNA. No virus contains both RNA and DNA. A virus is a nucleoprotein and the genetic material is infectious. The general viruses that infect plant have single-stranded RNA and viruses that infect animal have either single or double-stranded RNA or double-stranded DNA. Bacterial viruses or bacteriophages, viruses that infect the bacteria. are usually double-stranded DNA virus. The protein code called capsid, made of a small subunit called capsomere, protect the nucleic acid arranged in helical and polyhedral geometrical forms. Virus causes disease like mumps, smallpox, herpes, and influenza. AIDS, AIDS in humans is also caused by a virus. In plant, the symptom can be mosaic formation, leaf rolling and curling, yellowing and vein curling, dwarfing and stunted growth. Viroid. In 1971, Theo Daniel discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber disease. It was found to be a free RNA. It lacked the protein code that is found in viruses. Hence, the name is viroid. The RNA of viroid was of low molecular weight. Prions. In modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormal folded protein. The agent was similar in size to viruses. These agents were called prions. The most notable diseases caused by prions are bovine spongiform encephalopathy (BSE), commonly called mad cow disease in cattle, and its analogous variant, Cobb-Jacob disease (CJD) in humans. Lichens. Lichens are symbiotic associant, that is, mutually useful association between algae and fungi. The algal component is known as mycobient and fungal component as mycobient, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi, and fungi provide shelter and absorb mineral nutrition and water for its partner. So close is their association that if one saw a lichen in nature, and one would never imagine that they had two different organisms within them. Lichen are very good pollution indicator. They do not grow in polluted areas. Thank you so much.